Hey everybody, Mr. Murphy here. I'm going to continue with chapter 2.4, which is all about lipids. So lipids are hydrocarbons. They are primarily composed of carbon and hydrogen atoms. They do have some oxygen, but overall the big point is going to be the number of CH bonds contained within. They are insoluble in water, they are nonpolar, and because of all those covalent bonds, and they don't have any sort of hydroxyl groups and things, or they don't have many hydroxyl groups hanging out there, the distribution of electrons is much more even between all these carbon hydrogen bonds than they would be between, for instance, the very electronegative or comparatively electronegative oxygen and hydrogen that we see in carbohydrates and other molecules. When they're close together, there's a weak but still significant van der Waals force. Talked about this in an earlier section of the chapter, just that, yes, it's very weak, but it is present, and therefore we do see some clumping, or rather, we do see the clumping and association of non-polar objects together. So lipids will clump together. Now, these store that energy between these bonds. And so when we talk about lipids, we talk about them as these long-term energy molecules. The only reason why they're long-term compared to carbohydrates is just because our bodies and the cells of most organisms don't have a super immediate way to turn these bonds into direct ATP. And instead, for instance, in our bodies, we rely upon basically a depletion of our available glycogen and glucose stores, and then our body turns towards using fats, lipids, to convert into glucose so that we can continue to give those to the mitochondria and continue the process of cell respiration. Getting a little bit ahead of ourselves, but just to understand sort of why we talk about these as long-term energy versus short-term. They're great energy-carrying molecules because these fatty acid chains can be very long. So a ton of CH bonds that have that potential energy stored up within them, that if they are broken down, can be liberated and used for a variety of things. So they also play structural roles. This is understatement of the year. They play a structural role in cell membranes. The cell membrane is 99.9% lipids. It's phospholipids. It's a phospholipid bilayer. So fats actually surround every single one of our cells, which can also be a pretty logical thing that we'll get into in our next unit. But if you think about it, if we're talking about nonpolar substances such as lipids, being these molecules that don't play well with water, then if we're looking for a way to create a barrier between the watery interior of a cell and the watery exterior of a cell, what better than a fat, which is not going to play well with either side and therefore keep them separated. So it also provides thermal insulation. Think polar bears and bears in general. Bears are a great example of insulation using lipids. So triglycerides are the polymer of lipids, and these are the combination of 3 fatty acid chains and a glycerol molecule. They're extremely nonpolar, which is a weird thing to say. They are nonpolar, and therefore they are extremely hydrophobic. So 3 fatty acid chains and a glycerol group, and we will see here the dehydration synthesis of a glycerol molecule and 3 different fatty acids. So again, we have the carboxyl group on the fatty acids this time, and the hydroxyl group on the glycerol. An H from one side and OH from the other forms a water molecule and allows the O on this side, it wants another bond, the C here wants another bond, they bond together, and you have a triglyceride. Now, what fatty acids actually attach, or in some cases different things attach, like a phosphate group in the case of phospholipids, that's how we get sort of the unique characteristics of different types of fats, much the same as what we see with the different characteristics of carbohydrates and disaccharides and things like that. Depending on what specific monomers are actually used can create a whole host of variations and differences in the characteristics of these things. Saturated fatty acids are ones that have all of the carbons within it. There's no double bonds. And so we call it saturated because it's saturated with hydrogens. Every bond that could be found between a carbon and a hydrogen is found because the carbons don't have any double bonds that they're sort of wasting on a bond between two bonds between carbon molecules. Unsaturated fatty acids, though, they do have these double bonds. And that's going to create a difference in the shape. So here we have palmitic acid, which is a saturated fat, and you can see how it stacks sort of evenly here next to each other because it has this very straight shape. Meanwhile, linoleic acid, or linoleic acid, has these double bonds, and what that causes is kind of a kink and change in the shape of the fatty acid chain, and they don't stack as well, which actually leads to a whole list of characteristics of what we find with saturated versus unsaturated fats, and it's very logical and chemically based in terms of why saturated fats like animal fats tend to be solid at lower temperatures like room temperature, because they have this much more regular shape. They can stack together and solidify much more easily than unsaturated fats, which have a very irregular shape and therefore require a much lower temperature in order to become solid. You can stick olive oil in your freezer and it will solidify, but it needs a much lower energy, or think about that in reverse. It doesn't need as much energy to remain a liquid because of that irregular shape. So they're amphipathic, which means they have a hydrophilic end and a hydrophobic tail. That's individual fatty acids. Phospholipids use this to their advantage and create this hydrophobic tail end, and then the hydrophilic end, which is the phosphate group that is bound to the glycerol. So in an ordinary triglyceride, if we just jump back here, this whole thing is going to be nonpolar because there's not a significant polarity to glycerol and there's no polarity to the fatty acid chains. However, in the special case of phospholipids, we see basically a change to that third spot on the glycerol that's instead a phosphate group, which is hydrophilic, and allows for the unique characteristics of what we see with phospholipids and the phospholipid bilayer around cells. More on that in our next chapter. But here you can see what I'm talking about. That phosphate group allows for a hydrophilic region to exist on the on one side of the molecule versus the hydrophobic tail. So we'll get into that a lot more, but that's the phospholipid bilayer, and that's what allows for unique characteristics of how cells can separate their interior from exterior. So that's it on lipids, and in the next section we will look at energy. If you have questions, let me know.